Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. For those who know me, my name is Ammara Yuniar and I am a student of biology education in Semarang State University. Today, I will talk about the practical structure of animal epithelium tissue and its role in the body. I will start by defining epithelium tissue and why knowing it is important, then how to look at the epithelium, its part, characteristic, their function, and where they can be found in the body. Commenting of interest related to my submission, taken from animal tissue structure course, which has practical activities at the Multimedia Biology Laboratory FMIPA UNES, with a laboratory assistant named Mrs. Rafa and Mrs. Sabrina, guided by a practicum book in carrying out activities in the laboratorium, and a practicum report book were completed after the practicum. As for the provision of preparation, microscopes and computers are available in the laboratory, so students don't pay anything. And every time borrow a preparation for practicum only by writing down the borrower's identity. Okay, have you ever imagined how the epithelium tissue structures that make up the organ in the body? Or know that such a small tissue has such an important role in the body? I, as a biology education student, had the opportunity to do a practicum to find out the epithelium tissue structure, and I will share it with you. Epithelium tissue is a tissue in the form of cells with a tight package, without cell space, separated by a membrana basalis, on this case is basement membrane, from the underlying tissue. The function of the epithelium tissue include for protection, preventing water evaporation, absorption, excretion, and sensory receptors. The composition of the cells that make up the epithelium tissue based on the cell layer, it is divided into epithelium simplex and epithelium complex. But in this case, I will talk about epithelium simplex. Okay, the purpose of practical structure of animal epithelium tissue first is to recognize the different types of epithelium and then to know the shapes and function of the epithelium. Okay, that covers the definition of epithelium tissue, and let's move into objectives. The objects that used in this practicum are epithelium squamosum simplex on preparation of rainbow means capsule, then epithelium cubodium simplex on ovarium primary follicle preparation, epithelium cylindricum simplex on duodenum endothelium preparation, and the last is epithelium pseudocomplex on trachea preparation. Okay, next is the method. The method used in this practicum is qualitative method with observation using a microscope. Observation steps of the MOTIC BA210 microscope program as follows. First, the computer is turned on. Open the MOTIC-net application. Second, the microscope is connected to the switch. Then turn on the light. Then, the microscope camera is turned on by pulling the button. The preparations are placed on the object table and clamped. Then the edge of the object table is adjusted by rotating the macrometer. After the object is found, it is focused by rotating the micrometer. Finally, objects can be observed through a computer display. Okay, next I will show you how the detailed and interesting the tissue that make up the organ in the body are. The observation obtained from the microscope are as shown in the following figure. The following rows indicate the type of epithelium in the preparation. Okay, for the first, This is epithelium chromosome simplex in Bowman's capsula of the ran. This is the capsula Bowman in here. So this is the epithelium. Again, then this is the epithelium cylindricum simplex on the endothelium of the duodenum. Okay, the third is epithelium cuboideum simplex in the ovarium primary follicle. This is the primary follicle and this is the secondary follicle. But the epithelium cuboideum simplex is on the primary follicle. This is a small one in here. Okay, the last is epithelium pseudocomplex on trachea. Okay, in here. Okay, let's move on and discuss about characteristic, function, and where the epithelium can be found. Okay, for the first, we talk about epithelium chromosome simplex that has the characteristic flat shapes, only one layer in here, tight, thin, and the nucleus, in here nucleus is centered. The function of this epithelium is to protection, regulate substance that enter the underlying tissue and lining blood vessels. And this epithelium can be found in parietal lining of Bowman's capsule of the glomerulus of the ran, walls of arteries and veins, and walls of alveoli. Okay, and then is epithelium cuboid simplex that has the characteristic cube shapes. Only one layer in here, tight, and this is thicker than epithelium chromosome simplex. Has polygonal or honeycomb shapes, and the nucleus, in here nucleus is centered. The function of epithelium is to protection, absorption, secretion, and transport of substance. And this epithelium can found in thyroid gland follicles, ovarium surface, carinal epithelium, and renal tubulus. Okay, the third is epithelium cylindrical simplex that has the characteristic cylindrical or elongated shapes, tight, only one layer in here, and age greater than width. Age greater than width. Okay, and the nucleus is centered. The function of this epithelium is to protection, absorption, secretion, and active movement of digestive molecules. This epithelium can be found on inner surface of digestive tract or tractus digestivus from stomach to anus. And the last epithel is epithelium pseudocomplex that has the characteristic cylindrical or elongated shapes. And then as if layered even though the cell edges are not the same. In this part has the long cells in here, long cells, and short cells. And this epithelium has cilia in here and uneven nucleus. You can see this nucleus in the top and then one nucleus is in the bottom. Okay, and next for the function is the protection, secretion, and movement of respiratory mucus. And this epithelium can be found on the inner surface of the trachea, the inner surface of the bronchi, and the ductus deferens. Okay, the practical structure of the animal epithelium tissue has advantages, including first, multi-computer and microscope is in the good condition. And second, the presentation of the preparation objects by laboratory assistant is clear. And the last, testing the post-test question stimulates learning. But there are also disadvantages that are still found in this practicum. There are the preparation is damaged. For example, the cells are separated, so making it difficult for students to find objects. And then the presentation by the laboratory assistant is less efficient. She only explaining objects belonging to a certain group. And the last is expectation after practicum. Okay, this practicum can improve knowledge and skills of learning the structure of epithelium tissue. And then there is an update on preparation. So this is not damage for the student. So a student can see the object clear. And the last is improvement in presenting object preparation. So student can get the material properly or optimally. Okay, for the next session is discussion. Previously, I had distributed seminar materials to several of my friends, and they were unable to attend the seminar. But they shared the question via WhatsApp chats, and I will try to answer it. Okay, for the first question is from Adinda Sanifa. She is a student of science on senior high school. The question is, Hi, I am Adinda. I student a student of minoring in science at my high school. I have received epithelial tissue material before, but I could more detailed material after reading your material. I want to ask, then how should we respond to misconception in material during high schools and college? Okay. Hi Adinda, it's great to see your enthusiasm in learning the material that I provide. Yes, I once received on an explanation from my lecturer that indeed there were some misconceptions in the Indonesian education curriculum. I also only realized it after college. And I think, simply put, we don't have to wait for government policy to learn, right? As long as we know which things are right, then understand and implement them. Okay, we move on to the second question from Elmufida Eka Safitri. She is a student of Penyuluhan Pertanian Berkelanjutan on Pulbangtan, Yogyakarta. The question is, I'm not from a biology student, but I'm very interested in knowing your material related to the relevance of the benefits of studying the structure of epithelial tissue into this life and in the future. Can you show it? Okay, through this seminar, I think by knowing more detailed parts of the epithelial structure that participate in forming organs, we can be more optimal in maintaining their existence so that their function can run as they should. Okay, and then from Faza Nikmatul Maulia, she is a student of Pengembangan Informasi Masyarakat on UIN Walu Songo. The question is, what difficult did you experience when carrying out the practicum, saying how detailed the preparation you observed and studied were? Okay, actually among the practicums that I did in this semester, the practical structure of animal epithelium tissue is most fun. The practical make me fun because the microscope and MODICnet in the computer program are very easy to use. So preparation can be found quickly and precisely. Maybe it only takes about 1 minute find the object in focus. So it is helpful to quickly learn the parts. Okay, the last question is from Fernanda Debrari. She is a student of mathematics education on Semarang State University. The question is, Hello Mara, I'm Fernanda Dini. It's good to be able to get new knowledge from biology material. Okay, I'm going to ask what this says is likely to be experienced by the body to damage the structure of the epithelial tissue. It would be very scary if the epithelial structure in the body was damaged, because it means function in the body is as living cells, marked by the presence of a nucleus to protect every organ in the body. Of course, we have to maintain the presence of epithelial cells that make up our bodies while adhering to health patterns so that the organs around the epithelium can function properly too. Okay, I think it is enough for a discussion session. Finally, I would like to thank you, those who have helped in the structure of animal epithelium tissue practicum, so that practicum activities can run optimally and be useful. First, I say thank you for Semarang State University, Faculty of Mathematics and Science, Department Biology, Biological Media Laboratory, my lecturer in animal tissue structure. He is Insinyur Nur Tahayu Utami, Magister Science, and then Media Biology Laboratory staff, and my laboratory assistant, Mrs. Rafa and Mrs. Sabrina. Okay, to end this presentation, I also say thank you for your attention in listening to my material. I hope this is useful and we no longer just imagine how the shape and role the epithelium tissue should look like, but now we know and must take care of it so that it can function properly. Okay, that's all. The last but not least, wassalamualaikum warahmatullahi wabarakatuh.